Hello, my name is Arpita Bhattacharyya, and I just completed my PhD from Dr. Klapperich's lab here at BU. And today I'm going to demonstrate the fabrication of a thermoplastic microfluidic device using micro-hot embossing and thermal bonding. And then I'm going to demonstrate the isolation of nucleic acids using a solid phase containing silica particles, and the extracted nucleic acids can then be used for PCR amplification detection or any other diagnostic module.
Hello, my name is Dominika, Kulinski, and I'm a graduate student in the Biomedical Microdevices and Microenvironments Laboratory at Boston University. I will be talking about the formation of a carbon nanotube lysis monolith in a microfluidic device, and the formation of a solid phase extraction monolith for DNA extraction in a microfluidic device.
Today we are going to demonstrate an experiment on the production and application of microscale lysis columns and solid phase extraction columns within thermoplastic microfluidic channels. These columns can be used for bacterial cell lysis and isolation of nucleic acids from the cell lysate. The first step in the process is the fabrication of the microfluidic devices in cyclic polyolefin. We use a polymer which comes under the trade name Zeonex. Zeonex 690R has a glass transition temperature of 136 degrees Celsius and is UV transparent. UV transparency is very essential for the light-directed chemistry used in the fabrication of the porous monolith within the microfluidic channels. This microfluidic platform is fabricated using micro-hot embossing and thermal bonding.